The overall goal of this procedure is to evaluate the phenotype and suppressive function of natural CD4 positive regulatory T cells expanded from the peripheral blood of HIV-1 positive individuals. This is accomplished by first isolating the regulatory T cells by density gradient and flow-based cell sorting. In the second step, the regulatory T cells are expanded in culture. Next, the phenotype of the expanded cells is characterized by flow cytometry. In the final step, the function of the expanded Treg population is assessed by a T-cell suppression assay.
Investigating natural CD4 positive Tregs in HIV-infected individuals has traditionally been challenging, and in large part because of the scarcity of the population. The advantage of our technique is that it allows for the ex vivo generation of large numbers of these very rare cells.
The method demonstrated here may help to answer key questions in the HIV immunopathogenesis field by facilitating the characterization of the function and specificity of regulatory T cells in HIV disease progression.
Though this method can provide insight into peripheral blood regulatory T cells, it can also be applied to other body compartments, such as expansion of regulatory T cells from the gut-associated lymphoid tissue, a major site of HIV replication.
Begin by carefully transferring the blood from the ACD collection tubes into 50 milliliter conical tubes for a final volume of 15 milliliters of blood per tube. Then add 25 microliters of rosette